Good day everyone, my name is Keishi from Group 3. Now I'm going to summarize the topics that we have learned in 14 weeks under the course of Molecular Biology and Conservation into 6 categories. The first category is lab technique. There are 4 processes in lab techniques. The first one is extraction, followed by PCR, gel electrophoresis, and sequencing. In extraction, we have come across three types of techniques. That there are C-tap, phenol, chloroform, and Kaizen. In PCR, we have learned 3 steps. The first one is denaturation, followed by annealing and extension with 7 magic reagents such as DNA template, primers, magnesium, and etc. I think you guys should know that. In gel electrophoresis, there are examples of the gel such as starch, agarose, and pitch. In sequencing, there are 2 methods such as Sanger sequencing and next generation sequencing. Next, we proceed to category 2. That is processing DNA sequence. How do we do the alignment of the DNA? We can do it by manually or by computer software. By manually, we can make the DNA alignment from homology, and by computer software, there are bioedit and mega software. That's all from me. Thank you.
Hello guys, I'm Hongkawai. Next, I would like to talk about the next category, which is the analysis. The analysis we're doing is the phylogenetic analysis. First, we have 2 type of methods, which is distant matrix method and a character-based method. In distant matrix method, we have label joining, UPGMA, while the character-based method, we have maximum parsimony, maximum likelihood, and Bayesian inference. And next, after this method being conducted, the tree generator will verify by bootstrap. Usually we will run 500 bootstrap or 2000 bootstrap to make our tree more reliable. And next is the markers. There are 2 types of markers, genetic markers. One is the dominant marker, and another one is the co-dominant marker. RIPD is a dominant marker, while FLP microsatellite is a co-dominant marker. In co-dominant markers, this marker able to differentiate the heterozygosity of one or particular alleles, while dominant marker cannot do so. And next is the gene deposition. In the lecture, we have learned that there are 2 places for the gene to deposit, which is the gene bank and the bowl. The gene bank is repository, while the bowl is repository and data management. That's all.
Coming up next is the case study. For the case study below, I have prepared several sketches, and then the first one is the phylogenetic relationship for the taxonomy. As you can see, both one is not the main point is not banana, but it's humans and the monkey, and then they are not equal and they are not in the same group as the fish and the dolphin, because they are from a different group, and hence they are differentiated according to their own characteristics. The second ones will be the molecular clock. The molecular clock talks about the differentiation of the specimens and the taxonomy based on the fossils and their times and the mutation of rates. As you can see, the baby, and when after it experiences mutations over time, it becomes the Wolverine, the X-Men. The third one is the phylogeography. The phylogeography talks about in the maps, and as you can see inside the maps they got several colors, and this shows the distributions of the different animals or the different groups of taxonomy inside. The fourth one, phylogenetics diversity. is very simple. The picture tells everything. You can see the phylogenetic diversity is differentiated based on diversity, richness, functionals, and the evolutionary. The difference of the evolutionary is based on the stick. On the stick, the longer it is, that means that the diverse it is. The fourth one, this one is much more simple. Phylogenetic signals. The signals is depending on the same groups which share the same characteristics. The more characteristic they share, it means that the more similar they are. And if they inside the same group, the same species, they exhibit different characteristics, that means that the signal is last. These two, one is right, one is last. That's all. Now proceed to the next technique.
Hello. Okay, now I'm going to talk about the task that we have done from week 1 to week 14. Okay, the first task is this one over here. Why is it placed over here? Because the first task is based on the whole tree. We took an article and we read the article for more than 10 times and we highlighted all the words that we don't know and we do not understand. So it's placed here. Basically, the task is for the whole course. For the second task, it's based on abstraction. For the second task, from the article we have read, we identify the abstraction method they have used and from where the abstraction method, from where they take the tissue or the cell. For example, for amphibian, they have to take the toe clip or the tail clip. Then for the task 3 is the PCR, which is we select the genes most used in all the articles. So all of us have uploaded what gene is mostly used in the Padlet in Smart UNS. We can go to the website to have a look. From there, we find the most used gene and we conclude and we summarize what is the gene used the most. For example, mitochondrial gene is found to use the most because it's abundant and easy to be studied. For the task 4 is the gel electrophoresis. They are given 3 different gel electrophoresis, A, B, and C, and we have to differentiate what are the differences in these 3 electrophoresis. And we are given the controls, the positive controls and the negative controls, and we have to use all the controls and all the bands to show and to explain why is it different. Okay, for the task 5, alignment. For the alignment, we did alignment using the bioedit software. So we align all the sequences and we uploaded it in the smart tool. For task 6 is a phylogenetic tree. We use the mega software to edit all the trees and we come up with 4 trees, which is the maximum likelihood, maximum parsimony, neighbor joining, and UPGMA. From the 4 trees, we select the best tree and we use that as a consensus tree and we put all the numbers of the bootstrap there. For the last task is the task 7, which we did last week. The task 7 is basically the whole article that we have collected from all 40 of us, and we concluded them into a small piece of paper. We concluded what is the problem and what are the issues and what are the use of molecular biology in conservation. That's all. Thank you.
Hello everyone, my name is Shalini. Now I want to explain about tutorial. Basically, tutorial, there are altogether 4 tutorial sessions in this sem for this course. Basically, the tutorial helps us guideline. It's a guideline for us to do the tasks and assignments better. For example, in tutorial 1, Dr. Liew has explained us how to do the task 2, task 3 and task 4. And in tutorial 2 about task 5, how to use the software, the bioedit software, and how to download and the usage of the software. And basically in tutorial 3 is we learned about task 6, how to do the task 6, basically how to analyze the data according to the phylogenetic analysis, and how to use the software, the mega software. And in tutorial 4 is basically about we combine all together our 6 groups journal and discuss about the objectives and the outline and major issues of the conservation. So in conclusion, tutorial helps us to do our tasks and assignments better, and that's all.
Okay, let's move on for our predictions in our final examination. The first prediction is the 7 PCR reagents that have been needed to conduct the PCR reactions. The second prediction is the techniques, the lab techniques, the laboratory techniques, which starts from the DNA extractions until the DNA sequencing. what are the protocols and what are the procedures and what are the reagents used in all these steps. And what are then next prediction is the 5 factors in the phylogenetic tree constructions, which is taught us by our Dr. Liew. What are the 5 factors? The phylogenetic relationship, the phylogenetic diversity, the phylogenetic signal, and the molecular clock, phylogeography. All these 5 factors might may be asked. He may be asked us to elaborate all these steps, all these factors. And next prediction is how to construct the phylogenetic tree, the construction of the phylogenetic tree, which Dr. Liew taught us in our tutorial. And last prediction is the experimental design, which have been taught us in our last week of the lecture, week 14th. What are the aspects needed for the experimental design? That's all. Thank you.